All right, today we're going to be talking about the blood and the different components of the blood. So, blood is composed of three different things. Here on this top layer, we have what is called plasma. And plasma consists of mainly water and proteins. We have the buffy coat. And that is all of the white blood cells. And then here we have the red blood cells, or we also call it hematocrit. Now, this is used more in a clinical setting where you calculate a hematocrit level. So a normal hematocrit level is about 45%. So then we have this. We're going to talk about a little bit of the composition. So in a hemoglobin cell making up those red blood cells, we have these four sections. We have 2 alpha and 2 beta. And in a part of each of these, we have a section of heme. And the body can break down the globin without an issue, but heme, it has to go through a different process. So heme turns into biliverdin, which is a bluish green substance, and biliverdin can then turn into bilirubin, and albumin can transfer to the liver. So this is through a process called heme oxygenase. Biliverdin gets turned into bilirubin through biliverdin reductase. All right. So the process works. Draw a liver here. I'll label it because that does not look like a liver. Okay, the liver with the small intestine. Weird looking kidney. Okay. So we'll start. We have bilirubin that comes into the cell. And what attaches are these glucuronic acid substrates. They bind to it to make conjugated bilirubin. And this comes through the common bile duct. So this is a component of bile. So this enters the small intestine and gets turned from the conjugated bilirubin to urobilinogen. And then that has two paths that can go up to the blood and then eventually into the kidney, or it can continue in the small intestine. They go through the colon and then eventually out the rectum, but this is called sterobilin. Which then contributes to poop. So, or this urobilinogen can either go right here and recycle back to the liver, or it can go out with the urethra and be excreted as urine. So that's how we get rid of heme in the body. Alright, what we're now going to talk about is clotting. And clotting in the body, what do we call this? Hemostasis. So we're going to start out and draw a blood vessel. It consists of a bunch of cells. And we have, on top of the cells, we have this basement membrane. So, when we have something that disrupts this membrane, inside the cell we have all of these platelets here in the cell. What we also have these what we call VWF. And within the platelet, we have these binding sites. And so what happens when we do have this open part of the wall? Say you got a cut, say something happened. These VWF, they'll come and they'll bind right here. These platelets will come and they'll attach. And they'll just be activated just right here, right where it's broken. And as these come by, they will knock it off. As they knock them off, the next ones that are attached will then be activated. And these have a different couple characteristics about them. They have different granules. So what they have granules contribute to calcium. Increase in calcium. There is dense granules, which those contain ADP. We have TXA2. And we also have the alpha granules. The alpha granules consist of VWF, fibrinogen, factor 5, and factor 8. And so what activates this clotting process is this TXA2 and also ADP contribute to this. And this TX2, it's a G protein. Alright, and what this does is it actually starts what we refer to as the clotting cascade. So the clotting cascade has 3 different pathways. It has the intrinsic, the extrinsic, and the common. So the intrinsic pathway consists of factor 12 to activate then. So it goes factor 12 to factor 12A. Then from there, it takes factor 11 to factor 11A, 9 to 9A, and then 10 to 10A, and that starts the common process. So then we have factor 10 to 10A, and then it jumps to 2, which is called thrombin. And it goes to 1A, this is fibrinogen. Sorry, just 1 is fibrinogen. Going to 1A, which is fibrin. So the extrinsic pathway takes number 7A, which requires vitamin K. And this takes it all the way to 10A. And this is activated by collagen, which are found within the basement membrane up here. There's lots of collagen right here. And then this is activated by vasopressin, which is another name for tissue factor. Also another name for factor 3. And then there is a couple of drugs that will impair this. This has warfarin, and Gumden. We have heparin. The tests to figure out these levels are PTT in intrinsic and PT in extrinsic. And so the way this works, we call it the clotting cascade because when we're using what's very vital in this whole process is this common pathway, which you can see is at the tail end of it. We have thrombin, fibrinogen, and fibrin. And what fibrin do is when we are clotting, they're super vital in connecting these platelets together. They need a glue, and that's that fibrin. And so in order for us to make a lot of that, we need this clotting cascade to start, and it just keeps moving down the path until we get there to the bottom with fibrin, thrombin, and fibrinogen, which are all vital in our process. What we're lastly going to talk about is how we destroy coagulation, because we don't want to just have all of our blood cells coagulate and stop us from some living. So the way we do that is we destroy fibrin. And so what we have here is we have plasminogen goes to plasmin. So that fibrin mesh uses this TPA which are endothelial cells. So, as we have more TPA, and more plasmin, it results in destroying the fibrin mesh and reducing our coagulation. Alright, that's all that we have to talk about today. Thank you for watching.